When we picture a decaying, abandoned building, I think the first thing we look at is the structure.
Right, the load-bearing wall.
Exactly. We look at the load-bearing pillars, those massive concrete walls, and we instinctively assume that, when those start to crumble, the whole building goes down. We assume it's a structural failure.
It's definitely the most intuitive way to think about systemic collapse. I mean, you assume the biggest, most visible pieces must be the ones breaking to cause that kind of catastrophic damage.
But imagine you actually walk into this decaying building to inspect the damage. You get down into the foundation and you find out the load-bearing pillars are actually perfectly fine. They are totally intact, just doing their job. Instead, you discover that the building is collapsing because the custodial staff and the electricians have suddenly armed themselves, barricaded the basement, and started setting fires from the inside out.
That is a wild image, but honestly, it's perfectly accurate.
Right. That terrifying image is exactly what is happening inside the aging human brain. I mean, we have spent decades blaming the load-bearing pillars, our neurons, for age-related cognitive decline.
We really have.
But a landmark 2023 paper from the journal Cell just proved that the real uprising is coming from the brain's support staff. Welcome to the deep dive, by the way. Today, we are completely deconstructing the architecture of the aging brain.
And this paper, it's titled Molecular and Spatial Signatures of Mouse Brain Aging at Single-Cell Resolution. It fundamentally rewrites our understanding of what happens to our brain cells as the clock ticks.
It really does. Because for decades, the focus in neuroscience was, it was almost exclusively on the neurons.
Yeah, they are the star players.
Yeah.
They're the cells that actually fire the electrical signals that make up our thoughts, our memories, everything we do.
Okay, let's unpack this structural map before we look at the fires being set. Because mapping a mammalian brain over a whole lifespan, that is an incredibly heavy technological lift.
Oh, absolutely. The researchers looked at the frontal cortex and the striatum of mice across 3 distinct ages. So juvenile at 1 month, young adult at 6 months, and old in 21 months.
Got it. But to figure out exactly what was happening inside those brain regions, they couldn't just, look through a standard microscope.
No. They needed to know what the individual cells were doing at a molecular level and exactly where they were doing it. So to get they used a technique called single-nucleus RNA sequencing, or snRNA-seq.
snRNA-seq. Okay, I've seen RNA sequencing compared to, running a whole piece of tissue through a blender to see what genes are active.
That's a classic analogy, yeah.
Right, so you get this nice smoothie of genetic data, but you don't know which specific piece of fruit contributed which flavor. So single-nucleus sequencing changes that, right?
Exactly. Because instead of blending the tissue, they extract the intact nucleus from hundreds of thousands of individual brain cells.
Wait, just the nucleus? Why not just pull out the whole cell?
Well, the reason they use just the nucleus is that brain cells, especially neurons and glia, they have these incredibly long, complex branches.
Oh, right, the axons and dendrites.
Yeah, if you try to pull the whole cell out of mature brain tissue, you literally just rip it to shreds.
Yikes. Okay, so that doesn't work.
Right. But the nucleus is a sturdy little vault, and inside that vault is the RNA. RNA basically acts as the messenger, reading the DNA instructions and telling the cell what proteins to build.
So by sequencing the RNA of an individual cell's nucleus, you get its transcriptomic profile.
Exactly. You learn precisely what genes are turned on or off in that specific cell at that exact moment in its life.
So if I'm tracking the analogy here, snRNA-seq is like having the ability to intercept and read every single text message being sent in a major city.
I like that, yes.
You know exactly what people are talking about. You know someone is texting, I'm putting out a fire, and someone else is testing, I'm fixing a plumbing leak. But the problem with just intercepting the messages is that you have absolutely no idea where the people are standing when they hit send.
You lose the spatial context entirely. And in biology, tissue architecture is everything.
Location, location, location.
Exactly. Where a cell is physically located dictates its function, its local environment, and who its neighbors are. A cell screaming fire in the basement means something entirely different than a cell screaming fire on the roof.
Which brings us to their second and arguably more revolutionary tool, MERFISH.
Earthfish.
It sounds like a colorful deep-sea creature, but it actually stands for multiplexed error-robust fluorescence in situ hybridization.
Try saying that 3 times fast.
No thanks. But what's fascinating here is how it solves that GPS problem we just talked about. It's a spatial transcriptomic tool, right?
Right. Instead of extracting the nucleus out of the brain, Merfish leaves this ultra-thin slice of brain tissue completely intact on a slide.
Okay.
Then the researchers wash the tissue with these fluorescent probes that are designed to bind to very specific RNA molecules. In this study, they designed probes for 416 specific genes.
Wow, so over 400 specific targets?
Yeah, a mix of cell-type markers and genes that we already know change with age. By imaging these fluorescent dots under high-powered microscopes, they can identify the cell type, see exactly what genes are active, and plot its precise physical coordinates within the brain's architecture.
Okay, let's do a quick recap for you listening just to lock this in. So going back to the texting analogy, Merfish is basically like having the exact GPS coordinates for every single phone in that city.
Exactly.
So now you don't just know that someone is texting the word fire, you know they are standing in the basement of the building right next to the main breaker box.
You have the what and the where.
Right. So by combining these two tools, the text messages and the GPS coordinates on roughly 400,000 individual cells, they created this unprecedented high-resolution atlas of how the brain's cellular landscape shifts as the animal ages.
It is a massive achievement.
And this is where we get back to those load-bearing pillars, the neurons.
Right. And when they mapped the neurons across the 1-month, 6-month, and 21-month-old brains, the findings were honestly genuinely shocking in their lack of drama.
Lack of drama?
Yeah. The spatial organization and the overall abundance of neurons remained incredibly stable across the entire lifespan of the mice.
Wait, really?
Really. There were no massive die-offs. There was no catastrophic reorganization of where the neurons were located.
The structural pillars were holding up just fine.
They were fine.
But wait, this completely flips the script. If the neurons aren't dying off in droves, why is aging so famously associated with memory loss, slower processing speeds, and cognitive decline? Like, what is actually happening to the neurons if they aren't collapsing?
Well, it's not that the neurons experience absolutely zero changes. They are accumulating wear and tear. The researchers found that as neurons age, the genes they upregulate, meaning the genes they are actively turning on more than they used to, those are mostly related to dealing with oxidative stress and maintaining mitochondrial function.
Okay, so the mitochondria, the powerhouses of the cell, are basically sputtering.
Yeah, that's a good way to put it.
They are generating more metabolic exhaust, and the neuron is just working overtime to clean up that exhaust and stay alive. They are tired, but they aren't losing their core identity.
Precisely. They are still functioning neurons in the exact same physical locations. The profound, landscape-altering changes the massive shifts in gene expression and cellular identity. Those happen almost exclusively in the non-neuronal cells.
The support staff.
The support staff.
Which brings us to the glial cells. Historically, glia were just thought of as the glue holding the brain together, right? That's what the word means.
Right. But we now know they are active, dynamic managers of the brain's ecosystem.
The oligodendrocytes, the astrocytes, and the immune cells like microglia.
Yes. And when the researchers looked at the snRNA-seq data for these specific cells as the mice aged, it wasn't just a story of general wear and tear or oxidative stress like with the neuron.
What was it?
It was a massive upregulation of genes associated with aggressive, inflammatory, and immune responses.
Let's pause and summarize this real quick. So the neurons are just getting a bit worn out, but the support cells are actively mounting a severe defense. But who are they fighting? If there's no outside infection, why are they suddenly producing cytokines and complement proteins? Why is the support staff panicking and pulling the fire alarms?
That is the million-dollar question. And to figure out exactly what triggers the panic, the researchers zoomed in on the specific cell type experiencing the most drastic, complete identity shift in the aging brain, the oligodendrocyte.
I love talking about oligodendrocytes because their physical structure is so wild.
They are really unique.
Yeah, for those listening who might not be familiar, these are the cells that manufacture myelin. They reach out with these long arms and physically wrap thick, fatty lipid layers around the axons of our neurons. They essentially provide the physical insulation for the electrical wiring in our brain, which allows nerve signals to travel incredibly fast without leaking out.
It's brilliant engineering.
And because wrapping an axon takes so much energy, they also provide crucial metabolic support directly to the neuron.
They are absolutely vital for maintaining the integrity of the brain's circuitry. Now, the sequencing data revealed something that had never been seen at this resolution before. Oligodendrocytes actually exist in different states depending on the animal's age.
Okay, how so?
Well, in the juvenile mice, the 1-month-olds, Almost all the oligodendrocytes were in a state the researchers designated as oligo 1. Then, by the time the mice reached young adulthood at 6 months, that entire population shifted to a mature state called oligo 2.
Okay, that tracks perfectly with basic biology. The brain is developing, the wiring is getting insulated, and the cells mature.
Right. But then, in the 21-month-old mice, the elderly mice, a brand new state emerged, a state that was practically non-existent in the younger brains. They classified it as oligo 3.
Oligo 3.
And this wasn't just an older, slightly more tired version of oligo 2. It was a radical transcriptomic shift. The cell completely changed what it was doing.
And here is where the Murfish GPS data just completely flexes its muscles. Because these oligo 3 cells, they weren't just scattered randomly throughout the entire brain, right? They were highly concentrated in one specific vital anatomical region.
The corpus callosum.
Right.
This is the massive superhighway of white matter in the center of the brain. It's a thick band of millions of nerve fibers that connects the left and right hemispheres, allowing them to communicate.
So why there? Why does the corpus callosum become ground zero for this new oligo 3 state?
Because it is the most densely packed region of myelinated axons in the entire brain. It is high-traffic, high-maintenance real estate. Oligodendrocytes there are constantly working to maintain those fatty myelin sheaths.
That makes sense.
As the animal ages, that myelin naturally experiences wear and tear. It starts to break down, releasing lipid debris. Normally, the local cellular ecosystem cleans up that trash. But with age, the system gets overwhelmed. The oligodendrocytes in the corpus callosum become severely stressed by this accumulating damage, and that stress physically alters their gene expression, shifting them into the oligo 3 state.
Wow. And the chemistry of this oligo 3 state is actually alarming. When they looked at the genes these specific cells were upregulating, 2 massive red flags jumped out, C4b and IL-33.
Yes, those 2 are huge.
So I recognize IL-33 from some immunology reading. It's interleukin-33, which is a pro-inflammatory cytokine.
Exactly.
It's basically a biochemical scream for help. When a cell experiences severe trauma or necrosis, it pumps out IL-33 to summon the immune system to the site of the damage. But C4b is a complement protein, right? Doesn't that essentially paint a giant eat me sign on the cell membrane?
That is exactly how the mechanism works. The complement cascade is a crucial part of the innate immune system. When C4b tags a cell or cellular debris, it acts as a homing beacon for immune cells to come in and phagocytose, or literally eat and clear away the tagged material.
So you have these stressed oligodendrocytes in the main superhighway of the brain pumping out a siren to summon the immune system and simultaneously tagging themselves and their surroundings to be attacked and cleared.
It is a recipe for localized chaos.
Okay, let's pull all this together to make sure we aren't losing the forest for the trees here. We used to think brain aging was just neurons slowly dying. But what this map actually shows is that the insulation on the brain's main electrical wiring is fraying.
Right.
And the cells maintaining that insulation in the corpus callosum, they get so overwhelmed by the structural breakdown that they transform into this oligo 3 state and they start blaring an inflammatory fire alarm.
That is the core mechanism of the aging brain right there. The white matter physically breaks down, and the support cells trapped in that breakdown enter a highly reactive state, releasing intense distress signals.
So, a question for you listening. If you were the brain's immune system and you suddenly heard this blaring biochemical alarm coming specifically from the main central breaker box, how would you respond? Because the brain has designated first responders for exactly this kind of situation. Enter the microglia and the astrocytes.
Oh yeah, these are the heavy hitters of the glial world. Microglia are the resident immune cops of the central nervous system. They are a type of macrophage. In a healthy young brain, they exist in a resting or surveying state. They use these long, spindly branches to constantly feel around the brain tissue, looking for foreign invaders or dead cellular debris to prune away.
And what about the astrocytes?
Astrocytes are these beautiful star-shaped cells that act like the ultimate paramedics and maintenance workers. They manage the blood-brain barrier, they provide nutrients to neurons, and they keep the chemical environment perfectly balanced.
Here's where it gets really interesting, because the cops and paramedics of the immune system aren't just reacting blindly to getting older. This is a highly coordinated, geographically targeted chain reaction.
Right.
When microglia and astrocytes detect those inflammatory signals, like IL-33, they undergo a drastic morphological change. They pull in their branches, their cell bodies swell up, and they enter a highly aggressive reactive state to fight whatever is causing the damage.
And Murfish allowed the researchers to watch this mobilization happen in physical space. In the young brains, the microglia were resting evenly throughout the tissue. But in the 21-month-old brains, they became heavily activated. And while there was some general activation everywhere, there was a massive, statistically significant spike of extreme activation clustered right in the corpus callosum.
The exact same spot where our oligo-3 cells are screaming for help.
Precisely. And the astrocytes showed an even more spatially distinct signature of panic. In old mice, they became intensely activated near the pia, which is the outer protective surface of the brain near the ventricles, and drastically, intensely activated right in the white matter of the corpus callosum.
This is where that spatial transcriptomics data just absolutely shines, because the researchers didn't just notice that these angry immune cells were in the same general zip code as the panicking oligodendrocytes. They literally used the Murphish coordinates to measure the physical distance in micrometers between the activated responder cells and the oligo-3 cells.
They mapped the biological causality in physical space. It's incredible. They found a direct, highly significant correlation between how activated a microglial cell or an astrocyte was and its physical distance to those inflammatory oligo-3 cells.
Wow.
The closer a microglial cell was to an oligo-3 cell, the higher its activation score.
It's like looking at a satellite heat map of a forest fire. The closer you get to the source of the spark, the more firefighters you see swarming the area, and the more aggressive their tactics are. The oligodendrocytes in the white matter get stressed by the lipid breakdown. They release the IL-33 and C4b alarms, and the immune cops and paramedics rush to that exact location, entering a highly aggressive reactive state that floods the area with even more inflammation. It creates this vicious, localized hotspot.
The white matter is literally on fire with immune activity, driven purely by a breakdown in internal cellular homeostasis.
Let's do another quick recap. So the white matter becomes this massive hotspot for glial activation, entirely driven by localized breakdown.
Yes.
But that raises a massive conceptual issue. Anytime we talk about profound immune activation, cytokine storms, and inflammation in the body, it sounds exactly like what happens when we get sick.
It does.
If aging looks like a massive inflammatory cascade, is getting old just the biological equivalent of getting an infection? Are the mechanisms actually the same?
It is the most critical question to ask when you see data like this. So to test whether aging is just a generic inflammatory response, the researchers designed a brilliant control experiment. They took a group of young 6-month-old mice that do not have these oligo-3 cells or this age-related white matter breakdown, and they injected them with a substance called LPS.
LPS stands for lipopolysaccharide. It's a large molecule found on the outer wall of certain types of gram-negative bacteria, like E. coli. When you inject LPS into an animal, the immune system detects it immediately and thinks, oh no, we have a massive systemic bacterial infection. It triggers a severe, acute inflammatory response, essentially simulating sepsis.
If we connect this to the bigger picture, comparing the LPS-injected young brains to the normally aging old brains allows the researchers to strip away the generic noise. It shows us what a standard panic response looks like, and contrasts it with the unique, intrinsic signature of the aging process itself.
And the results of that comparison were fascinating because the initial reactions actually looked very similar, right? Both the bacterial simulation and normal aging triggered widespread release of pro-inflammatory cytokines. Both events caused the microglia and astrocytes to completely abandon their resting states and activate.
But the crucial differences emerged when they looked at where and why that panic was happening. LPS causes a systemic, uniform panic driven from the outside in. Because the fake bacterial signal is coming from the bloodstream, when you look at the astrocytes in the LPS-injected mice, they are heavily activated right alongside the blood vessels. They are fortifying the walls, fighting off what they perceive as an outside invader trying to cross the blood-brain barrier.
But aging is an inside job. There is no external bacteria trying to get in.
Exactly. Aging causes a strictly localized panic. That specific, intense activation of microglia and astrocytes clustered tightly around oligodendrocytes deep in the white matter of the corpus callosum. That spatial signature was completely unique to aging. The bacterial infection did not cause that hotspot to form.
The LPS sickness was a generalized, citywide alarm ringing because an invader was at the outer gates. The aging signature was a localized, intense structural fire in the central breaker box of the building, caused by the building's own faulty wiring.
It proves definitively that brain aging is not just a general slowing down of the system, and is not just a generic inflammatory response to living in a dirty world. It is a highly specific, structurally localized event driven by internal cellular breakdown.
So, to summarize the journey we've just been on today, what we've learned fundamentally changes how we view the timeline of our minds. Brain aging isn't a tragic story of our neurons quietly fading away into the night. It is a highly specific, geographically targeted inflammatory uprising led by the brain's non-neuronal support staff.
Perfectly said.
It is an event concentrated in the white matter superhighways, where the cells responsible for insulating the wiring get so overwhelmed by the breakdown of their environment that they call down a swarm of immune cells, inadvertently setting the very tissue they're trying to protect on fire.
This raises an important question, though, one that could entirely shift the future of medicine.
Oh.
Yeah. For decades, the pharmaceutical industry has poured billions upon billions of dollars into therapies designed to target neurons. We've tried to keep them alive or help them clear up plaques or stimulate them to fire better, all in a desperate bid to prevent age-related cognitive decline and dementias.
Right.
But if the cognitive decline is actually being triggered by this localized inflammatory fire in the support cells, does that mean future therapies should stop targeting the neurons entirely?
Wow.
Should we instead be designing drugs that specifically target the IL-33 immune pathways of the oligodendrocytes?
Yeah.
Can we treat age-related cognitive decline by simply calming down the white matter?
It is a profound, provocative thought to mull over. If we want to save the decaying building, maybe we need to stop worrying about reinforcing the pillars and we start focusing on calming down the maintenance crew in the basement before they burn the whole place down.
Yeah.
Thank you for joining us on this deep dive into the microscopic geography of the mind. As you go about your day today, maybe take a moment to look at your own support systems, the hidden networks, the routines, the people that keep your life running in a whole new light. Because sometimes the most important story isn't happening on the main stage, it's happening in the background. Until next time.